All right, gang, here we go. Let's do a little Unit 1.2 test review. So I'm going to walk through everything that would be on your test. This should match up pretty nicely with your review guide that we put together for you as well. So first things first, we talked a little bit about what is called algor mortis, which is time of death. You're going to have to be able to read a graph like this and be able to interpret um, when the person has died based on their body temperature, right? So one of the key things, if I pull up a pen here real quick, is if I said that the patient or the deceased body was at a temperature of about 29 degrees Celsius, you should try to figure out how long ago the person has died. So if you use this y-axis here that says temperature in degrees Celsius, 29 degrees Celsius would maybe be about right here or so. You're going to take that and you're going to drag it over to your standard curve here that we have, and then kind of come down. And wherever it is on the x-axis should give you an approximation of how long ago the person died. So this might be like 11-ish, maybe 11 and a half hours ago, something along that line. So that's one of the key components. You're going to have a graphical analysis piece for that element of algor mortis. Remember, we did a little activity that was around that. The other thing that you have to know, so that's algor mortis, right, which is the time of death. You're also going to have to look at things like rigor mortis. So remember, rigor mortis is connected to, I'm going to just pull this up right here, muscles and the stiffening of muscles, because your body is no longer making ATP. And so what happens is, if you remember, we had those myosin fibers and the actin fibers that were sitting over each other. Hopefully you remember this. And there's like little kind of arms that reach up and grab that actin. And those little arms of the myosin, when they grab up and grab that actin, they're going to squeeze these actin fibers closer together. Well, what happens at death is you're no longer making ATP, which prohibits the myosin from releasing from the actin. So the muscles get stuck in the position that they're in. So hence the rigor means stuck or fixed, and mortis means death, right? So your muscles get fixed into that position, happens within the first couple hours, and then after about 24 hours, 48 hours, the body starts kind of decomposing, and then it releases based on the decomposition. Okay, so no longer making ATP prevents the muscles from releasing the myosin from the actin. That's the cellular component of it. Liver mortis is the pooling of blood that we see because the blood isn't being pushed around by the heart, right? So we basically have pooling because of gravity. So if the body of the person was laying, if they were laying on their back, right? So it's a quick rendition of person laying on their back. Here's their arm here like this. What you're going to see is the blood will start pooling on the bottom half of their body because gravity is pulling down this direction. If the person was somehow fixed in a chair, right? So person in a chair, right? Then you might see there would be pooling in the lower portions, maybe the bottoms of their feet, their buttocks region, maybe their hands. Maybe you start seeing stuff in the lower part of it. Depending on how they are oriented to the earth and gravity is how you're going to see that pooling. That would just give you a snapshot of how the person possibly died within the first couple hours, right? Because once that person dies, then the blood will no longer be moving around with the force of your cardiac muscle. It'll be stuck wherever it is and start moving down towards that gravity. We also looked at things like entomology, which is the study of insects and how they're connected to the human body and decomposition. So remember, once you die, your normal immune responses to prevent such things halt. And so insects will find this as a food source. A lot of animals can as well. The bacteria that's in you and on you, we talked about that, how the microbial components will start eating you, and that's what causes some of the bloating and the smell that you get from dead bodies is the bacterial byproducts. It's not just because the muscles are decomposing, it's because the bacteria are eating you. But also the insects. So watching or understanding how an egg hatches into things like its larval state, and through the multiple what are called instars or levels of the larva developing into things like a pupa, where it might do a metamorphosis and move into its adult form. Understanding that about each insect that might predate or use the human body as a food source can give you a time period that that body has died, right? So if you see lots and lots of maggoty types of things, well, then those are larvae and that they're past the egg stage. So it might be multiple hours. It depends really on lots of different conditions. We talked about how not only is it important to understand where the body was found, right? So that plays a big part in the algor mortis, if they're outside or inside, whether they're clothed or unclothed, the humidity that plays a part, the wind speed, all those things play a big part for the entomology as well. Because if you have a dead body that's on the inside of the house, you might see a totally different group of insects than if the body was outside of the house, right? Out in the environment, the woods or something like that. So that plays a big part. Climate and the environment all play a big component to this time of death. And so how have scientists figured out a way to collect data on all these different variables? And that's where the body farms component comes in. So body farms are these scientific endeavors at multiple different research facilities where they put dead bodies into the environment with varying different variables, right? Like having a bed over a body or putting them in sand versus soil. And all those elements play a big part in trying to get baselines of data to help people figure out, other scientists figure out how long a body has previously died, right? So that's the time of death elements. We did that for the first week or two of this section that we were taking this test on. And then we looked at the digestion and how it connects to toxicology. So you have to know the elements of, once again, the tube of you, right? Which is basically your mouth to your anus. You have to know the elements that are part of the tube, but you also need to know what are called accessory structures. So accessory structures are the things that help feed in some materials, I shouldn't say feed, dump in materials into the digestive tube there that can aid in this digestive process. Because remember, the goal of digestion is to take large molecules, get them small so that they can get absorbed. That's the big goal of this. So up here at number 1, where the mouth is, you have teeth that can help do some of the grinding and increase the surface area of your food. But you also have an accessory structure right here, which is a salivary gland. You got multiple paired glands, 3 different pairs of salivary glands, and they're all going to dump in this solution called saliva. And saliva, yes, has lots of water, but it also has enzymes that can help us with things like carbs. I'll start breaking down some carbohydrates. It then moves into this tube here that's all lined with smooth muscle, and that's the esophagus. The esophagus is going to do this involuntary contraction, squeeze all your food down to number 3, which is your stomach. Now remember, this is just a general overview. We can go into a heck of a lot more detail about the digestive tract in other time periods, but we're just doing the generic component. In the stomach, we'll get things like acid that are dumped in there, specifically hydrochloric acid. That's going to really drop the pH. The pH in your mouth might be like 7 to 8-ish. We're dumping all the way down to things like 1 to 3, depending on how your body is reacting at any given time. So it gets really acidic, and that helps loosen the bonds of our food, make it a little bit easier for the enzymes to work on. But notice we really haven't done a whole lot of absorption yet. We then move on to the first part of a really long piece of your digestive tract. Not necessarily large diameter, but long piece, and that's the small intestine. So number 4 here is the small intestine, running out of space. Right, so that's the small intestine. And the small intestine we'll also have two other accessory organs that are going to dump in. Number 10, which is paired with this little sac structure. Number 10 is the liver, and on the underside of the liver, you'll have the gallbladder. That gallbladder will store what the liver is producing so that it can inject it into the digestive tract, specifically the small intestine, when it needs it. And that liver is going to secrete a substance called bile. And bile helps emulsify fats. Basically, it takes little flat globules and makes them tiny so it's easier for enzymes to work on it. And then you get to the pancreas, number 8. And the pancreas is going to release just a ton of digestive enzymes, tons of them. And these digestive enzymes are going to run the gauntlet. They are going to break down carbohydrates to the small sizes of monosaccharides. They're going to take fats and turn them into little teeny fatty acids. They're going to take proteins and turn them down to teeny tiny amino acids. They're going to take nucleic acids like DNA and RNA and stuff like that and break them down all the way down to nucleotides so that we can get them absorbed. So in the small intestine, you get a majority of your bigger components of digestion, but also your absorption. So this turning the large to small and absorbing, almost all of that is happening really down in that small intestine. You would not be able to survive without that small intestine. And it's also super stinking long, right? It's about 3 times the length of your normal human height, right? So if you're 6 foot, it'd be about 18 feet long of small intestine that's all coiled up in there in that abdominal cavity of yours. And then we finish up the journey with the large intestine. And the large intestine is just large based on its diameter compared to the small intestine. So you got that little niblet of a piece there, that's the appendix, but this crinkly piece coming around this direction, that is all large intestine. And it finishes out, another synonym for the large intestine is the colon. So you have like ascending, transverse, descending, sigmoid, colon, all those play a part into what we call the large intestine. And then it dumps out rectum, anus. And so that large intestine houses tons and tons of bacteria. I guess I should write this on here. So number 5 is the large intestine, LG intestine. And it houses a ton of what we call our microbiota, right? So bacteria that is not harmful, more than likely it's beneficial. And they will help you process up some of the last bits of food, help you get a little bit more absorbed vitamins and things of that nature. And in turn, they're eating all the food products that you have. It also helps make your feces smell not so pleasant. But the large intestine will also help with your last-ditch effort to remove water, because water is so vital for your survival that if you're freely releasing all that hard work of drinking the water, then you could possibly die. So the large intestine works really hard at removing the last little bits of that water that you might have in your food sources. Okay, this connected to the toxicology part was, well, there's a lot of things that we can take into our body that can be pretty harmful, and it's a job of forensic toxicologists to look at what we have in digestive tracts or in the bloodstream to see if that was the source of why the person was either not behaving well or died, right? So we gave you a chart very similar to this with your forensic toxicology, and your goal was to use how indicators react with known substances to match up how those same indicators react with an unknown. So you're going to take the information from the unknowns and match them up with the knowns. Okay, the knowns in this example are acetaminophen and cocaine, heroin, oxycodone, and ecstasy. So if you look at unknown substance 1, it shows that there's no change for indicator 1. And that would match ecstasy, and that would match, sorry, my pen's not activated here. That would match ecstasy, that would match heroin, would match cocaine. You're basically doing a puzzle with this, right? And so then unknown substance number 1 with the second indicator produces a cloudy white, which is like the cocaine. It's not like the other substances. So now we've got 2 checks positive for cocaine. And then you look at the last one, it says yellow and bubbling. Ooh, so we got yellow and bubbling as well. Interesting. So maybe what we have here is two substances combined into our unknown substance. So right now, preliminary results of these three indicators would say that unknown substance 1 could possibly be cocaine and heroin together, right? We might need to run more diagnostic tests to figure that out. But at least with this, we have a pretty good idea that it's reacting similarly, these indicators, to the known substances of cocaine and heroin. Now, unknown substance number 2, we get bubbling for oxycodone, right? So that's a star there. We get no change, which matches heroin, and then we get a white solid, which matches acetaminophen. So what could we possibly put down for this one? Well, it's very possible. Oh, there's also a no change with acetaminophen, but there's also bubbling with acetaminophen. So where we have our check marks out, right? It could be oxycodone, but it didn't match up. It didn't get a green yellow solid here. It didn't get a no change on our unknown 3. Right? Same thing with heroin. We have some marking yes, some no, but acetaminophen is a knockout. It matched on all 3 accounts with all 3 indicators. So chances are unknown substance 2 is acetaminophen. Okay? All right. On to the next part. We started talking a little bit about the nervous system. So that's where the eye diagram comes into play. And so with the eye diagram, you need to know a majority of these elements here. We've walked through all of them. I'll kind of walk through them real quick because you're watching the video right now. So number 1 is the cornea. And we even related that back to death. That's a way for us to figure out if a person's eyeball is not functioning properly, because the cornea should be clear when it's healthy. If the person dies, that can cloud up, right? That's an indicator. Or if the eyeball does not have the proper nutrients, it can also start clouding up. Essentially, the eye is dying within possibly a living human body. Number 2 is the aqueous humor. So I'm just going to put aqueous humor. It's basically a water substance that keeps that front part of the eye inflated and in the right shape. Number 4 is showing a gap or a hole, and that's the pupil. Number 5 is this element here, which is the color of your eyes, which is the iris. So that's going to help control how much light comes into your eyeball. So there's a muscle connection and a nervous system response that connects in there. Number 5, we weren't too worried about. These are drainage ports to revitalize the nutrients, because that aqueous humor and this other substance back here, which is the vitreous humor, all need to be recycled. It can get kind of gunky, so your body is constantly recycling it. Number 8 and number 6 are connected together. These are the ciliary bodies, and they are muscles that help hold and pull and relax this muscle, or this structure number 7, which is the lens, and that lens helps focus the light for you. I already said that the 14 is the vitreous humor. It's a jelly-like protein that helps keep the eyeball in its shape, and it allows light to pass through. When the light does come through the pupil and through the lens and gets focused, it should focus a majority of the image right there on number 12, which is the fovea centralis. But you still get reception on this disc right here in general. And that whole disc, that spot, the fovea centralis is within it, is number 11, which is the retina. So the retina is this cup, the inner cup of your eye, that you have the rods and cones, right? We talked about those, receiving information of light energy. We didn't really talk about number 10. 10 is the vascular tunic. It's the blood supply that comes in. And number 9, we did talk about. This is the sclera. This is the whites of your eye. It's a part of what's called the fibrous tunic, or the tough wrapping around your eyeball. Number 15 is the blind spot, because there is no rods or cones in that location, so there's no reception of information. And number 13, we connected to our brain via the optic nerve. So that's how all the information is getting sent to your brain for your brain to process. Speaking of brains, another element of our units is looking at things like this, which are MRIs. Oops. MRIs, magnetic resonance images. So this is showing soft tissue, the different densities found within soft tissue, specifically the cerebrum of your brain, that's the upper part of your brain. So this top MRI would be of a normal brain. It's got good density in the main center part. You can see the corpus callosum is nicely connected, so we have both hemispheres nice and attached. We get a little bit more fattiness out near the edges. You can see there's more fattiness down here as well, but all the brain is pretty much connected together the way it should be. In the second one down here, this is for CTE, or chronic traumatic encephalitis, and that is a lot of damage, progressive over long periods of time. We're seeing wearing away of some of that soft tissue. We got a lot more gaps than we should. The only other MRI that you might see is if you saw a traumatic brain injury, TBI, and you would see large swaths of dead tissue in different areas of the brain, but it would show up as a nice white clump within that brain. Okay, you do also have to know the different sections of the brain. And so we're primarily focused on the cerebrum, but I give you all of it just because, hey, why not? It's science, right? So the eyeball, for reference, would be up near the front here. This is where the eyeball would be, and it would connect in. So this is the frontal lobe, which is going to be primarily focused with logic and control of muscles. But the control of muscles mostly comes out of this ribbon right here, which is the primary motor cortex. And number two, the next part, is the parietal, which is going to do a lot of sensation components. And specifically, a lot of your touch sensation is going to come in at number 6, which is the somatosensory cortex. Right? Moving onward, move down to number 3 here. This is going to be the temporal lobe. That temporal lobe is going to help you with your ability to hear, your ability to taste, to comprehend language. So there's a lot of integration that happens in this temporal lobe, especially to locations like the memory component part of your brain, which is inside the brain. If you split the brain open, the hippocampus and other important things for memory are located right underneath this temporal lobe on the inside, or the surface of the temporal lobe on the inside there. Finishing out the stuff that you definitely need to know for the test about the brain is number 4, which is the occipital. And the occipital is where you're going to process vision. And all of your important vision comes into that back location. Number 7 is the cerebellum. Number 8 is the pons. Number 9 is the medulla oblongata leading into the brainstem. All of those are part of the autonomic components, or the automatic components of your nervous system. They do a whole host of vital stuff, but it's considered part of your what's called your hindbrain. Controls swallowing and breathing and heart rates and things of that nature. Very important that you have those things, but not necessarily important for what we're doing, at least right now in PBS. Okay. We then boiled it all the way down to a neuron, which that's what this is. Pull up my pen again. So this is a neuron, which is one of the main functioning units of your nervous system. We said that the signal only moves in one way. It'll be moving from the left-hand side on this diagram to the right. It is electrical on the inside, but let's hit at it, right? So on these little spiky branches off to the left here, all these are what are called dendrites, and this is where one neuron is going to connect into this neuron. So you might have little synaptic buttons or terminals touching in here from lots of different sources. Remember, they can be sending in a positive signal or a negative signal, saying to this neuron, hey, you need to fire, whereas some of them are saying, hey, don't fire. This section right here, number 4, would be the cell body, or soma. So all that signal gets routed down the cell body, and it gets down to this section right here, which is the axon hillock, who will make, I'm anthropomorphizing here, who will choose whether or not this neuron actually fires. And when it does fire, if it does, it's going to jump as it moves down. Saltatory conduction between these little 2 spots, which is called the nodes of Ranvier. Now, it's important to note that this whole unit is an axon. So the node of Ranvier and the axon helic are part of it. And then we have these myelin sheath wrappings by neuroglia like Schwann cells or oligodendrocytes. So that's myelin sheathing. And that's like a fatty insulation to make sure that the neuron's electrical impulse is moving the correct way at the proper speed. We finish out our signal moving down here to number 5, which are the terminal, the axon terminals, or the buttons. And that's where the electrical impulse stops being electrical, and it transforms itself because this cell can make proteins and fats and things of that nature and excrete them out of the terminals here. And those little chemicals are called neurotransmitters. So they're secreting these little chemicals into the gap between one cell and another. So we get, if we zoomed in, we'd see something like this. Here's the button, right? Here's the little neurotransmitters in it. The electrical impulse is coming down here, gets down to the end, and these little chemicals get pushed out into this gap. This right here is cell number 1, and this would be cell number 2. And these little neurotransmitters are going to activate number 2 of the next neuron, which is the dendrites, maybe, of the next neuron. So these little neurotransmitters might be saying, hey, you need to fire, positive, 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 positive. And some of them that come in might say, no, you need to not fire, right? So depending on what kind of signal, what kind of neuron, we can get different types of neurotransmitters activating or deactivating another neuron that follows that. Okay, we finished out this unit talking about the cardiovascular system. So remember, the cardiovascular system, at the heart of it is the heart, right? And this is a muscle that receives blood but also can push out blood. So it is all numbered out here. You need to know the flow, like what comes next in the series, but also how the heart works through that process. So I'm going to start with 7 and 8. So 7 and 8 are the vena cava, and they are veins, so they're returning blood back to the heart. Remember, arteries away, veins towards. So the veins are bringing back the blood. They tend to have lower blood pressure, not always, but they tend to have lower blood pressure. So 7 and 8 would be vena cavas, and they're both dumping their contents in here. So if I did like a woo, it's coming down, woo, right? It's dumping into 1, and 1 is the right atrium. Wait, right. Yeah, remember, it's patient perspective. So this is the patient laying on your surgery table. This is their right side on the left-hand side of the diagram, okay? Remember that atriums are above, ventricles are below, so you get this really cool kind of shape coming out, right? There it is. Atriums on the top, ventricles on the bottom. This side would be the right, this side would be the left. That's what we're seeing generically here, right? So then the right atrium will push past A, which is a valve. That is the tricuspid valve. Right, right? It's in there. Right is within tri. So it's going to be on the tri, because D over here is going to be the bicuspid valve, right? So we move from the right atrium down to the right ventricle. I'm just going to put RV. And then the right ventricle is going to push it up through B, which is a semilunar valve, because it is half moon-shaped. C is also a semilunar valve. Blood moves up here into 5. So 5, right and left, is moving away from the heart. So these would be pulmonary, because it's got to get oxygenated at the lungs, pulmonary. And this is away from the heart, artery. So this is the one place where an artery has deoxygenated blood. It gets oxygenated, comes back via 9 here. So these are pulmonary veins. Write it once here. And they would be oxygenated veins. They come back to 3, which is top of the heart. This is the left side, so this can be the L A, or left atrium. It dumps down through the bicuspid valve into 4, which is going to be the left ventricle. Moves back up through semilunar valve and hits the big kahuna of pressure, number 6, the aorta. And aorta will push that blood all throughout the body, and then once it gets used up, it comes back to the vena cavas. And now we've gone through the entire heart in one full process. And that's kind of what you have to do with the flow of blood as you go through it. You need to know how to walk through that process. So starting maybe, if I said somewhere like, hey, you started in the right, no, let's start left. Let's start with the left ventricle. You need to be able to say what comes next, what comes next, what comes next, all the way back to it ends up back at the left ventricle, because it is very figure 8-like, the blood flow, right? It's very figure 8-like. So what else do you need to know as far as blood vessels are concerned? You need to know what the different types are and how they're anatomically similar / different, right? So vessel-wise, there's arteries. Remember, arteries are away. They tend to have lots of flex because they're going to have an increased amount of pressure in them because they tend to be right after the heart, and the heart squeezes and increases that pressure. Veins, conversely, are going to be towards the heart. It's going to have less flex, but there's low amounts of pressure, low pressure. So what they need are these things called valves. We talked about valves, that they're one way. So the veins get the blood back to the heart by the other skeletal tissue squeezing those veins. So if we made a vein here and we had a one-way door, we put a couple one-way doors here. Let's say we're just talking about your gastrocnemius, which is your calf muscle. It squeezes maybe here, right? The blood is going to want to push and go both directions, but it can only move upward because this door right here, this door is going to slam shut, so the blood pushes upward. The muscle squeezes here, and we continue the directionality. And this is why, if you remember sat for too long and then you finally stand up, you'll notice that where your socks are on your leg, you'll start seeing like a squeeze in from where the socks are because the blood was having a hard time getting back. This is also why if you tend to patients in the hospital, you want to elevate their legs so the blood drains back to their heart via gravity because they're not getting up and walking around. The last of the vessels that you need to know are the capillaries. And the capillaries are the smallest diameter, they are the most numerous, and this is where you're going to get all of your exchange. Exchange of what? Exchange of oxygen with CO2 in the lungs, exchange of oxygen with CO2 in all your cells, the exchange of glucose, your food source, with all the cells that need the glucose and the waste that comes from it. This is your exchange zone, your capillaries, and that's why they're the most numerous. They're all over your body. Anywhere that your body needs nutrients or to get rid of waste, you're going to have capillaries. So it doesn't matter if it's your heart, it doesn't matter if it's your brain, it doesn't matter if it's your kidney, capillaries are everywhere. And with that, I've finished the review session. Hopefully this has helped you a little bit. Walk through a majority of the diagrams. Feel free to go back and look at them. If there's any issues or concerns, by all means, reach out. You know I'm just an email away. I bid you adieu. Good luck with your studies. Thanks for watching.